complement activation pathways. There are three nonspecific, innate, humoral immunity pathways that use a variety of proteins called complement. It is a misnomer. Students often think that complement means one thing, but in fact there are complement components and three different pathways that often lead to very similar outcomes. These are some of our first-line defense mechanisms that provide protection from very early stages of infection. This is a chart that's essentially a phylogenetic relationship chart, starting with insects like Drosophila or fruit flies, going up through the various different types of animal sort of groupings like sea urchins, echinoderms, all the way up through reptiles, birds, and mammals. And if we take a look at some of the earliest forms of immune factors that provide protection from infection, and even some of the sort of very distinctively evolutionarily early types of organisms like sea urchins, we can see that it's complement and some complement type proteins. They're some of the earliest first-line immune defense factors. As I mentioned, complement is actually a series of proteins that interact. They have a variety of functions. So here we see two tables of functional protein classes that we can find in the various complement pathways. Some of them simply have the function of binding to the surface of a pathogen, like C1q and MBL. Some of them form active enzymes on the surface of the pathogen or the bacteria. Some of them, when we attach them to the surface of the microorganism, assemble into what we call attack proteins. And even others are mediators of inflammation and result in the types of symptoms we often associate with an inflammatory response. So a really wide range of functions for these complement pathways and components. Since we have individual protein components that are complement components, then we have to have receptors for them. That's the CR, complement receptors. So at any time you see a capital R in many of the abbreviations that immunologists use, try to remember that that capital R is almost universally referring to a receptor of some kind. So here we have CR1, complement receptor 1. And for complement receptor 1, we can see that the ligand, or the complement component that would bind to CR1, could be C3b, C4b, or IC3b. Notice that the types of cells where we'd find a CR1 would include not only the types of cells that we associate with immunity, like phagocytes, macrophages, neutrophils, also lymphocytes, or what's FDC, follicular dendritic cells. But for CR1s, we can also find those complement receptors on erythrocytes, red blood cells. Why do you suppose we would have a receptor for complement on an erythrocyte? We'll find out in a few more slides. The other types of complement receptors, CR2, 3, and 4, are generally found on cells we associate with the immune system. CR2, found on B lymphocytes and follicular dendritic cells. CR3 and 4 on phagocytes and dendritic cells. C5a and C3a receptors are found in cells lining various mucous membranes. We call them endothelial cells. Also on mast cells, as well as phagocytes, because C3a and C5a are going to be involved in inflammatory reactions. Notice that along with having the abbreviation CR1, 2, 3, or 4, these receptors also have additional designations called CD designations. So the CR1s are also called CD35s, the CR2s are CD21s. The CR3 and 4 also have additional designations. depending upon who's doing the discussion or what the point of view or when the discussion has been generated. Some of the CD designations are more recent additions, and some of the other abbreviations are actually older labels associated with these same receptors. So yes, immunologists do use a lot of abbreviations, and it can get very confusing. Just do the best you can, and eventually with more and more discussion, and we try to consistently just use the CR designations in our discussions for our class, but you will occasionally have to refer back to this table just in case a CD designation has been used, let's say in a case study, just to make sure what they're talking about, which receptor. For the three possible complement pathways, we have the classical pathway, the MB lectin pathway, and the alternative pathway. How do they differ? How are they unique? It's essentially called the attachment or recognition phase. All three pathways have unique recognition phases. For the classical pathway, we have an antigen-antibody complex that forms on the surface of a pathogen. For the MBL pathway, we have lectins that bind to the pathogen surface. And for the alternative pathway, that also can be called the alternate pathway, we simply have the pathogen cell surface. Those are the recognition phases, when we have complement components attaching initially, getting this whole process started, to a pathogen cell surface. Once that occurs, then we're going to enter into what we call the enzymatic phase. We have complement activation. Now we really start to get this process going. When complement activation occurs, there are three possible outcomes. The most sort of notable is killing of the pathogen. That, of course, is a truly desired outcome. So we prevent the establishment of an infection. The middle possibility, and also occurring at the same time, is opsonization of a pathogen. Complement can serve as an opsonin. An opsonin is any chemical that can be attached to the surface of a pathogen or microorganism and enhance or facilitate phagocytosis. So think of it as adding extra targets on the surface of something that we want a phagocyte to ingest or eliminate. That's what an opsonin is. It's going to enhance phagocytosis. And the third possibility is the recruitment of inflammatory cells to the site of complement activation. That recruitment often brings very beneficial cells to the site to eliminate the pathogen. Once we've completed the recognition phase, then we can enter into the enzymatic phase. Notice for the recognition phase for the classical pathway, we're going to have three different complement components: C1, C4, and C2. These are listed in the sequence because this is the sequence in which these complement components get activated. They were numbered according to historically when they were discovered. So C1 was discovered, and then C2, and then C4, and so they're not numbered in sequence, unfortunately. So we have the antigen-antibody complex, essentially landing on the cell surface. Notice that C1 actually is a multimer. It comes in various subunits we call C1q, C1r, and C1s. For the MBL pathway, we have mannose on the pathogen surface. Mannose is a sugar. And so mannose is the sugar on the pathogen, and then we have mannose-binding lectin produced in our bodies that attach to that mannose. So MBL, in fact, when we take a look and compare the structure of MBL to C1, there's some structural similarities, but they're very different proteins. MBL and MASP1 and MASP2, along with C4 and C2, are going to be part of the enzymatic phase for MBL, or the mannan-binding lectin pathway. And the alternate pathway, or alternative pathway, we simply have to have the pathogen surface, the complement component C3, and factor B and factor D. When any one of these three possible combinations occurs, Then we're going to generate an enzyme called C3 convertase. And that's what you want to zero in on. What are the similarities and what are the differences in the recognition phases and the enzymatic phases of these three distinctively different complement pathways? Once we've created the C3 convertase, then we have various outcomes. One of the outcomes, as we mentioned just a few slides ago, is essentially destruction or killing the pathogen. We are going to achieve that when we establish what's called a membrane attack complex on the pathogen cell surface. That's illustrated on the right-hand side of the screen, where we have a complement component of C3. It's a split product called C3b. C3b attached to the pathogen surface then triggers the deposit of additional complement components C5b, 6, 7, 8, and 9. That's going to help to form what we call a membrane attack complex. We'll discuss that in more detail in a few more slides. C3b all by itself can actually serve as an opsonin, which enhances the function of a phagocyte. The opsonin essentially coats or covers the surface of the pathogen, makes it much easier for the phagocyte to attach to those C3b structures on the pathogen surface, and then remove that pathogen or ingest it, phagocytize it. We also have split products that are produced in complement activation. Typically there are several different complement components, C4, C2, and C3, and C5, that are split into two pieces, a small and a larger component or subunit. The small pieces are often designated A, so we have C3A and C5A on the right-hand side, or left-hand side of the screen. And we also have C4A and C2A that are produced as split products. So these smaller components have different functions, different activities. So C3A and C5A are actually going to be mediators of inflammation. C3b can serve as an opsonin or trigger the formation of the membrane attack complex. So nothing is wasted. The natural process for activation of the complement components is to pretty much use every component to have some type of important function in nonspecific immunity. What's missing in this illustration is a second enzyme. So C3 convertase, which is the first enzyme produced, is critically important in all three pathways of complement activation. What's also important, but not illustrated on this slide, or in most slides actually, is the formation of the second enzyme called C5 convertase. We'll talk more about how we generate C5 convertase in a few more slides.